And wanted to say just up front, there are some images in the PowerPoint here that we don't have citation or copyright for. The intended use is educational, non-commercial. Just wanted to say that up front.
So I'm Dan Seifert, also with the Forest Service. I'm the Cave and Karst Coordinator for Forest Service Region 1. You can see Bruce and Justin's titles. They're both with the Forest Service's Washington office. So thank you guys for being willing to be here tonight. And I'm just going to do the intro slide and the closeout slide as it relates to the Forest Service Cave and Karst program. So thank you guys for having us. It's always a pleasure to do things with Northern Rocky Mountain Grotto. And yeah, with that, let's jump right into the next slide. Okay, so I just wanted to talk in general about caves and karst on the Forest Service on National Forest lands. The map is a map that shows where all the known caves are on Forest Service lands in the U.S. And this is known caves. This is what's in our official Forest Service database. And I think at one point, Johanna Kovarik, who used to be the National Cave and Karst Coordinator Program person, had mentioned that she thought about 40% of the Forest Service's caves were actually in that database. So, you know, you could estimate there's another 50 or 60% more caves than that 3,900 number. Of those ones in our database, 2,400 of them are designated significant. And, you know, that's a bit of a process. There are a number of criteria for designating caves as significant, and those are listed off to the left over here in these bullet points. One of the important ones is paleontological resources, so that's fossils, and that's what we're here to talk about tonight. And also, I just wanted to throw out there that a lot of the forests are still actively inventorying cave and karst areas, and we do work with cooperating groups such as Northern Rocky Mountain Grotto and other grottos of the NSS to help us get our inventories better and try to figure out where important cave and karst resources are that need a little bit of management, attention, or protection, or perhaps interpretation. And scientific research, such as looking at the fossils that are in the caves. So yeah, that's just a little bit about the national program, and I think with that, I'll turn it back over to Bruce and Justin.
All right, thanks, Dan. So one of the things you guys really want to learn about tonight is just identifying the bones that you find in caves. And oftentimes, as you can see here, there's lots of people going into caves. Some of them, like bat biologists, might know a few different types of skeletal anatomy, but they don't frequently include paleontologists who are trained to recognize not just bats, but also mice, bears, lizards, and all kinds of shells as well. So we tend to be very broad, although some of us do tend to focus in a little bit more tightly on different taxonomic groups. So most of the things we're going to talk about tonight are the types of fossils that you'll find lying in the cave, as opposed to those fossils that you can find in the cave walls. Now, these can be of multiple different ages, from millions to only, you know, hundreds or thousands of years old. And the younger they are, the more prone to being disturbed they are by our exploration of caves. So as I mentioned, we're going to be looking at a broad thing, a broad topic. There's lots of things that wind up in caves, wind up lying on the floors of caves. Bruce and I are vertebrate paleontologists. We tend to focus on bones. So we're going to focus heavily on vertebrate anatomy, because those are also going to be the most obvious things you'll run into when you see fossils in caves. Now, the next most obvious thing you're going to see is fossil poop. And that's usually from rodents, but also probably bats. But we can also find various snail fossils, clam fossils, anything that could live in water, other invertebrates, those would be in there as well. So it's very, very diverse types of things that wind up in caves. Okay, as Dan mentioned before, paleontology is one of those significance criterions for designating a significant cave. These are actually some of my favorite types of fossils. You've got Arctodus here, which is basically the largest land mammalian carnivore to have lived in North America, except for maybe something that lived about 65 million years ago, but we won't get into that because I don't know that those ever wound up in caves. But these bears, they do den in caves, although we don't know what Arctodus is doing hibernation-wise. We do know that black bears and brown bears frequent caves and wind up often dying during hibernation in those caves. So they look like Dan's background if you check that out, although not mounted as prettily as it is. Okay, so there's lots of different ethical things we have to consider when we're exploring not just caves, but also the paleontological resources. One of the things we'd like to not do, where we'd rather have you as first explorers in there not do, is really disturb the fossils. Now, I know that that's going to be tough as you're crawling through on your belly with your elbows and your knees, so it's, you know, within reason. Try not to disturb things. That context that we're going to get from looking at the spatial relationship of bones to other bones is going to tell us the story of how they got there. So the best thing you can do is try to get around it without disturbing it. Take pictures with a scale bar, or if you don't have a scale bar, you know, something with a measurement on it, a lens cap that you know, or a rock hammer, or even a pen would work just fine. A dollar bill is 6 inches, and we'll know what that means if we see that next to a fossil. So those are good things that you can often have with you, and just lay it right next to the fossil if you can, and take pictures. And then, you know, the best thing is taking notes. If you have time to take a few short notes, you can then make a really nice brief report on what you'd seen in the cave. A picture like this that you see here is going to be perfect. We don't need the scientific names. We'll figure that stuff out later. But if you take away something from this presentation, you ought to be able to break down at least what birds and rabbits look like relative to rodents, maybe relative to lizards, and then even the bigger mammals, which Bruce will talk about after I get done here. In fact, I've got a bigger mammal crawling behind me right now. My dog might bark if the neighbors show up, so apologies for that. But this is a basic report that we'd really like to see, okay? And it's going to help drive forward the research and give us a good idea of what type of age of deposit is wound up in this cave. And that's important because we can run into caves that have a lot of animals, which Bruce and I care about, but they might have been brought there by people. So there's a lot of different things that go into the cultural side of things. And once people get involved, we start handing off a lot of the responsibility over to the archaeologists. They're going to be concerned about how these various animals may or may not have been used in the cave and in the cultures that were using the cave. Now, we'll still participate doing identifications and things like that, but at this point, the law gets really touchy, and the archaeologists are specialists with dealing with the cultural law, so we let them deal with it. Clues that can give that away, there's some things that are a little bit more subtle, like lithic scatter. You might not notice that, and green bone breakage. Tool marks might be mistaken for rodent gnaw marks, but things that are dead giveaways are rock art, fire, and you know, if you recognize a human bone, which we're going to try to get you to kind of see the difference between what humans look like versus other similar animals, which a bear can look fairly similar, as you'll see, or something that's been modified for use. As you might see here, there was a concern that this might be a chunk of antler, which antlers are often used for flint knapping, along with this bison bone over here, and other rub marks in the cave over here as well. So how do the bones get there? That's one of the things that we try to solve as paleontologists. We're trying to tell the story of how these animals got there. Now, it may be a cave habitat for an animal. You know, some invertebrates that use it are centipedes, scorpions, and things that feed on them, like shrews and bats. They're going to live in these caves and hunt them in these environments, or potentially go out, travel, and then come back and use these environments as shelters like bears, wolves, and other animals do. Okay, they could be pitfall traps. You know, little bunny foo-foo hopping through the forest all of a sudden doesn't see a hole in front of him and just drops in and breaks some bones and can't get out. Now this happened in a fairly famous cave in Wyoming, Natural Trap Cave, and not just for small mammals like foxes and rodents and rabbits, but also big mammals like the giant short-faced bear, and even mammoths can fall into these kind of caves and sinkholes like this. And this particular type of deposition can be episodic or continuous. For instance, if you get generations upon generations of animals living in a cave, and if they're predators, they're going to be bringing bones in. So you might wonder, why do we have bison bones in caves? Bison don't use caves, and neither do horses. Mammoths do, but you have to have a really big cave for that. So the predators are then killing an animal or scavenging a carcass and bringing those bones back. And that's telling you a lot about the environment that's going on in that particular part of the world. And of course, we've got anthropogenic effects, which just means people caching bones for either tool use, potentially fire use, potentially for food resources as well. Okay, so we're going to look at a whole bunch of other types of context. We're going to be looking as to what kind of deposition is going on in the cave. Is it rapid? Is it really slow? If it's rapid, particularly if there's pack rats in there, the deposition is going to be relatively continuous, because that pack rat is not only bringing in bones, but also covering it with its urine and feces. So it's kind of collecting things for us. That's really great. But that has an effect on the preservation. So we're going to get different staining based on the types of water that is and is not coming into the caves. Whether or not the cave is dry or a humid cave is going to affect the texture of the bone and how it breaks down. So we've got some of these relatively clean looking bones here, like this carnivore down here. And then we've got some more broken up stuff that's covered in mud and probably pack rat poop. And then you've got a rodent bone up over here as well. So it can happen in different types of situations, and even become cemented, where even relatively recent stuff with the right chemicals in the water can be cemented in place by caliche, which is a carbonate ironstone, basically hardening over that, kind of like flowstone does. And in South Dakota, there are some caves where that's happened to horse and camel bones. So of about 200 and some odd thousand years. Okay, so one of the things that you're going to basically notice first when we find these bones is, figuring out what kind of element it is, what part of the skeleton it is. And rather than trying to figure out what kind of animal it is, which I'll let Bruce deal with in a little bit, we're going to figure out just what type of bones we're looking at. So here we're looking at metatarsals. These are the bones that make up the top of our feet just before the toes happen. And that's what they look like in a horse versus an elk. So you can see here a horse has one toe, and it's reflected by this kind of singular beam going through that bone. Okay, that's going to be the main bone in the foot. Elk also have one bone, but it's fused from two, so it's going to look like it has this groove and then two ends at the bottom. And that's frequent in other even-hoofed animals as well. Birds having three toes, they have something down here that looks similar but expanded upon. So you see a trend there. Now our upper thigh bones are going to look different, but you can get a good sense of scale. If you have something that's a few inches long, you know it's not human. If it's about a foot long, give or take, it's going to be something in the range that could be human. And then we're going to have all different types of bones throughout the body. Ribs are fairly easy because they're nice and curved. And then vertebrae are also very, very distinct. Now the problem is figuring out where these things come from can be changed based on the use of the limb in question. So here we have a diagram showing all the different arm bones. They all have the same names per arm bone, but as you can see, for each animal, they're going to be extremely different based on their function. So a human is going to have a fairly generalized long humerus, whereas pterosaurs and birds have mechanically advantageous humerus, as do bats, flying. So they're going to get a lot of movement out here with a lot of power being generated on the humerus here. Okay, so that's what we call homologous structures, and they can have, in this case, for our flying animals here, our flying vertebrates, they've all converged on the same function and some similar structures, but you can see that not everything is the same. Okay, and this can be modified by the age of the animal. We call that ontogeny. One of the best ways to look at that is we look at the limb bones and we see whether or not the shafts have fused to the ends. So normally, where you see these bones being separated here, there's going to be a cartilaginous pad that over the lifespan of the animal starts turning from cartilage to bone. And once it's completely turned to bone, we have a reasonable good idea of what age that animal is. So that gives us another clue as to what's going on here. And of course, you can see that these animals, as I've mentioned before, they have mostly the same types of bones. Now, what I'll go into real quick is that our birds and other what we call lower vertebrates outside of mammals, non-mammalian vertebrates, are going to have what we call dorsal vertebra over the rib cage. That means they all support ribs. Whereas in mammals, not all of our backbone between the hips and the shoulders support ribs. So we have our lumbar vertebra and the vertebra supporting ribs. Now, vertebrae are pretty distinct. You'll make them out pretty quickly. They're usually disc or cylinder shaped, and they'll have a lot of different protrusions going on here. So we have, in this case, the centrum facing the top of the screen would be where the skull would be oriented, with this beam right here, the neural spine, being the back of the animal. Then we have two transverse processes, the neural arch where the spinal cord passes through, and where each vertebra articulates with each other, the pre and post apophyses. It's going to be a little bit different in non-mammalian vertebrates. They're going to be much more complex. Their vertebrae aren't completely fused all the way, but they're also going to be much, much smaller. Okay, we have a couple different terms for the way that these bones interact with each other at the centrum. That column down here, that's not the neural spine or where the spinal cord passes through. So this, in its joining up with the one in front and behind it, it's going to have a different concave or non-concave structure. And we use big fancy words to tell other paleontologists and biologists what that means. So birds are particularly unique. They usually have pockets in their bones, and they have these saddle-shaped articular facets. That's what we call them when they adjoin another bone. So pretty complex. And of course, the ones right up against the skull are going to be particularly special. They're going to be differently shaped. This is an atlas. Atlas as in it's holding up the spherical head. Okay, so it's got this kind of cup-shaped structure to it. And then you're going to have the atlas over here. That's going to have what we call a dens. It butts into the atlas here, and this provides both the rotation right here at the axis and the back and forth movement for the atlas. Okay, so that then goes into the cervical verts, the thoracic verts here, and then we move, if we have them, into lumbar and the tail vertebra, which are going to be much more elongate and with less extravagant side projections. Okay, and this is where I'm going to turn it over to Bruce. He's going to tell you how to figure out which animal is which based on these various different bones. Perfect.
Thank you, Justin. I appreciate that. So this is kind of a whirlwind through various different vertebrate groups. What we focused on here really is vertebrae and limb bones, and there's a reason for that. You find them often. You picture any vertebrate skeleton, there are lots of vertebrae, there are lots of limb bones, there are a lot of ribs. However, ribs are typically a little bit difficult to identify. You might be able to tell fish versus mammal, something like that, but not very diagnostic. However, vertebrae and limb bones are. As you'll see, we're not going to talk about skulls and teeth till the very end, because actually that would be a talk in itself, especially of mammal. paleontology. So much work is done from skulls and especially the teeth. But fish bones, we don't often think, you know, cave deposit, maybe something on the coast. You wouldn't necessarily think about there being a lot of fish bones in caves in the North American interior, but in fact there are. They're obvious. The vertebrae are amphicoelous, meaning that they're cupped on both ends. They're usually very nicely decorated along the sides, the lateral margins. The spines are very thin. And these are the most obvious bones that you're going to see, although skull bones from fish, likely to be disarticulated, can be very distinctive. And obviously, depending on the location of the cave, fish bones in caves can be freshwater, marine, or both. And they're actually a lot more common than one might think. Here's an example from the Tennessee River Basin to give you a sense. We're not finding articulated things. You know, we're often focused on the charismatic megafauna, whether alive on the landscape or dead in the cave. Really the magic in most of the cave deposits is the small bits that you really have to sift for. And in this case, you can see from this cave in Tennessee, pike, catfish, perch, drum, gar, a lot of modern fish identified in this sample of micros, teeth, and various jaw elements. And just a real quick focus on catfish because they are common not only in cave deposits and throughout the fossil record from certainly all of the Cenozoic, whether it's the pectoral fin spines, these really neat bones that are underneath the gill arches called the clythra, dermal bones from the skull roof. They're just very robust, easy to preserve in the world of fish. Catfish are, so they turn up quite a bit. The family Icthaluridae, everything from bullhead to all the various kinds of cats. So I wanted to share that. Kind of lumped here, we're going to transition from fish into something with most of the time limbs, not always in the case of the snake, but notice with amphibians, reptiles, turtles. In the case of a reptile, the snake, notice the difference in the vertebrae right away. So you don't have cups on both ends, you have a ball and socket arrangement, what's called procoelous. Snake vertebrae are very distinctive. Obviously no scale here, but this is blown up quite a bit. This would be about 1/2 centimeter long perhaps. Again, in the middle, an example of a Canadian cave, lots of frog, lots of snake bones, limb bones, vertebrae from these caves. So a lot of really excellent ecological work can be done from these small micros. And turtles, obviously, going to be very different. I'm not certain about the rarity, or I would think that turtles are a pretty rare component in cave deposits, but they're certainly very common in the terrestrial fossil record. Birds, very common find in caves. In fact, some of the most fun finds, I think, in caves are bird bones, especially the limb bones, because they're very distinctive. They're diagnostic to genus or even perhaps to species in some cases, but it's very easy to say, hey, this family of bird is in this cave. The vertebrae are very distinctive. They're heterocoelous. They have this saddle shape on the front of the vertebrae with extra holes and air sacs. And in fact, their bones are hollow, which you may be aware of. Very distinctive part of avian anatomy is their cardiovascular system extends inside the bones. And looking at the wing here, you can see how distinctive the radius ulna humerus would be for something like a turkey vulture. Hind limb as well, very distinctive bones in birds. So if you find limb bones, chances are you're going, as a novice person with a little bit of effort and looking online, you can identify the type of bird, at least the family. And other unique things about birds, you may have, in the middle here, this is a series of essentially tail vertebrae into the sacral vertebrae, but in birds, the whole package is kind of welded together into this thing called the sensacrum. It's all the pelvic bones and the sacral vertebrae. People are forever picking these up in the field and saying, look, this looks like a skull. They're very busy elements, so very distinctive sacrum. Obviously, given the capability for flight in a lot of birds, the sternum and the clavicles or the wishbone are very distinctive things. So birds are fun in caves. They're easy to differentiate from other things. Moving on to rodents again, this one is, as it says, it's really a can of worms. It's a can of rodents. The diversity here is through the roof. The real magic of a lot of Cenozoic paleontology, and in fact caves paleontology, is having a rodent specialist, because these animals are very useful for ecological analysis, climate, relative age dating. All of these animals in this very diverse group, all the way from porcupine to beaver to good old neotoma, they breed quickly, multiple generations, numerous individuals. So lots and lots of the bones, typically that are screened from caves, are rodents. And again, the teeth really come into being able to really do paleo magic with the class Rodentia. Rabbit bones I single out specifically because they, again, are very distinctive animal, but they're often confused for bird bones. They're gracile, they're very lightweight, almost hollow, like birds, and superficially some aspects of the limb bones of rabbits do look like birds, so they're often confused. That's the reason that I include this slide here. And also to point out that the skull, especially of a rabbit, with this really incredibly loose network in the front of the nasal area of rabbits, is so distinctive. In fact, a good tip, one of the few mammalian groups you'll see with doubled incisors in the upper part of the skull. So one tooth tidbit there. And the vertebral column, this is kind of going back to the theme that look how well differentiated things are and how easy it is for a person. When you have it all lined up, you can tell cervical vertebrae, the neck vertebrae of a rabbit, from dorsals and lumbars and into the sacrum and the tail, all very easily differentiated. Can't go through mammals without mentioning bats. Their bones in some caves are obviously very abundant where the habitat is right. Many different species. The bones are distinctive in that they're very lightweight and fragile. In fact, hyaline, meaning they're opaque, or you can almost see through some of the bones because they're so thin and so delicate. May occur as isolated bone fragments or even single teeth. Again, the limb bones are hollow like birds, but very different in terms of anatomy, easy to differentiate from birds. And the dentition, classic insectivore morphology to the teeth. And I think this funny question, how many cervical vertebrae does a bat have? Maybe we'll come back to that. That's a mammal question. The answer is 7. Carnivora, huge group. You can read the list: weasels, badgers, otters, canids, felids, bears. The most distinctive attributes of carnivore dentition is shown to the right. It's this biting anatomy that both dogs and cats share, this overlapping shearing mechanism they have between their premolar and their lower molar, a carnassial shear, able to crunch bones or cut meat. So very easy to differentiate carnivores based on that attribute, but also the limb bones. Again, very often they have these very deeply set processes in the humeri, is what we're showing here, a bunch of forelimbs from badger, fox, saber-toothed cat, and a canid. And notice in all of them, they have these very deep pockets, the olecranon notch, where the lower limb bones insert. I'm going to keep going through this pretty quickly so we can leave time for questions at the end. Justin touched on some of this. Some of the most noticeable things that may be in caves, the macro stuff. The bigger it is, the easier it is to notice. The more likely we are to be intrigued and investigate some of these things. And although it would seem like to a person not used to or accustomed to looking at what does a cow versus a horse versus an elk versus a moose, what does it look like, the difference between perissodactyls, represented here by a horse, and an artiodactyl are quite distinct. Justin was showing you earlier how being single-toed, We see a lot of different features in the humerus, this big third trochanter on the femur, for example, the asymmetrical astragalus, these ankle bones, a lot of different features that make it easy to tell perissodactyls, in this case horse, from artiodactyls, which is really the diverse herbivore that we're likely to see in caves. Sheep, goat, deer, bison, bovids, on and on. And again, double-toed animal or split hoof. And that's reflected in their cannon bones and this notch in the bottom, the line up these fused metapodials, and reflected in the limb bones, lack of that big third trochanter and a symmetrical ankle bone or an astragalus here. So it's very easy to differentiate between perissodactyls and artiodactyls is the point here. And kind of going through the chain here, no disrespect to us, we humans, we are mammals. Our remains, our bony anatomy is obviously very similar to all other mammals in many respects. We have 7 cervical vertebrae, but our limb bones are uniquely human. Very good reason for that. We're bipedal. We have these very long arms and tool users and all those kinds of things. So I think the number one thing to point out on this slide is that it's not uncommon. There's a black bear femur at left. Notice the overall similarity to a resin or a plastic copy of a human bone on the surface, but on the other hand, look how much more robust the bear bone is. Look how incredibly gracile the femur and the upper arm bones of the humeri of humans are. So superficially similar, yes, but not really. And also extending into the hand and the paw of a bear, human hand at left, bear paw at right. So that's something we obviously want to pay attention to. If we do suspect that there are human remains, at that point it's time to get an archaeologist, state SHPO, or hopefully not, but maybe even a coroner involved as appropriate. And coming kind of to the end, like I say, we haven't said a whole lot about teeth, but teeth are, in mammalian paleontology especially, that's really where it's at. One example shown here in the photographs at bottom, a person really can differentiate between, although they're very closely related, a good old cow, a very old cow, and a bison tooth based on features like this isolated stylid that's in the tooth. And there are different patterns. Deer, they have this, what are called a selenodont condition, sort of these half moon shapes in the teeth. Beaver and elephants, of all things, share this feature, this tooth type called lophodont. There are some animals that have incredibly long cheek teeth, like this horse tooth, because they erupt throughout life and are slowly worn down. So I really can't say enough about teeth in one slide, but the reason we focused on the vertebrae and the limb bones, as it says here, there are lots of them. There's only one skull with teeth. It's very common in paleontology. We're often disappointed we find a beautiful skeleton, be it a mammal or a dinosaur, and we go through the neck and the skull is either missing or smashed. They're delicate. They're more air than bones. If we're lucky, we get teeth, but it's often more practical to identify the postcrania because there's just so much more to work with. The axial skeleton and the ribs can be a little more challenging, but the limb bones are especially helpful. So kind of tying a bow on this, and we want to leave some time for questions. We're running, I think, a little bit long here. But the element at right, based on what we've shown, the question we would get from somebody is, what is this animal? And the first thing that I would respond to a person with is, what's wrong with the picture? To me, there's no scale. I can sort of guess based on particle size, but it would be so much better if there were a ballpoint pen, anything in the picture to give you a sense of the scale of this bone. And although this says found to be humerus, it actually looks like this is a femur. I think anybody can pick out the fact that it has this nice femoral head And in fact, it's got this very distinct muscle attachment on the femoral head. I believe this is a rabbit, not surprising. To contrast that with what is this element? What's wrong with this picture? Again, no scale, but what is the animal? It's a mammal, and it's very easy to pick out that in fact it's a carnivore, likely a cat, because it has this very distinctive, or maybe I have that, Justin, you can chime in and correct me if I'm wrong, maybe a canid, where the olecranon notch here, basically where the elbow joint articulates long range of motion with the bottom of the humerus. This would be the bottom of the upper arm bone. This hole is
probably a wolf.
Yeah, dire wolf, very distinctive in canids, whether they're large or small, maybe even in Canis familiaris, but very obvious things we can key in on. So to kind of summarize, just a laundry list, and I'll kick it to Justin here at some point and see what his kind of parting thoughts are about this. We're talking about just, vertebrates, just tips to bones, not the shells and other things that may be in caves. Fish bones are obvious and more common in caves than we might think. Pretty easy to learn how to differentiate what a fish vertebrae is, at least the centrum, the spool part of the vertebra. And small mammal bones, that's really where the specialist is needed. The rodents and the rabbits, they're incredibly diverse, very common in caves. Bones of large herbivores may be found less common depending on the cave, but often found at much higher elevation than we would expect, or beyond tight squeezes, which often raises some head scratching. How did this get here? Bones and teeth of carnivores and mustelids are highly distinctive, easy to differentiate those from other mammals. And bird limb bones are very distinctive, but I learned quickly you have to be careful not to confuse rabbit bones for bird bones. They're also hollow and gracile. For folks that are interested, there's a wide range of things that are available online now. I'm not an obligate caves paleontologist, but when I was going through vertebrate anatomy for modern mammals and other creatures, there were a few references, hard copy references, like these Mammalian Osteology by Gilbert, and there's a companion, Avian Osteology by Gilbert, that were available hard copy, dating myself, going back to the late 80s, early 90s. Now online, there's a wealth of resources to help a person identify bones in cave deposits or whatever you want to identify. So I'll see if we're at the, I'm going to click one more. Dan, I'll let you, if you'd like to, kind of give us the takeaway message on the summary slide here. And then I would ask Justin if you have any other things you'd like to mention before we go to questions. So Dan, if you wouldn't mind.
Sure. Yeah. I mean, that's one thing with caves and cave paleontology is, the element of time. So a cave has overall a relatively stable temperature and humidity. You can preserve things for a long time. I also find that interesting with some of the pit caves where we've got, say like Natural Trap or some of the other smaller pits across Montana, where you've got accumulations of fossils that can span a long time period. So you've got multiple species that are showing up, multiple ages of critters. So that's an intriguing thing, is just the time factor in terms of what a cave can preserve and capture. So part of that is we just need to think about what we're doing in a cave. The old thing, watch your step, leave careful footprints, as Bruce alluded to earlier. So we just need to think about our impacts. If you are traversing through a cave for exploration, in addition to watching out for the formations on the walls, look for fossils. Try not to tromp on those. And if you do find fossils, try to get some photos of them with a scale. Try to let folks know what you found. Within the Forest Service, Bruce and Justin are pretty accessible. I've had pretty good luck emailing photos to a couple of the Forest Service paleontologists and getting feedback. So if any of you ever have questions, feel free to route it through me or Bruce or Justin, and we can try to help you out. And also we talked about there could be a cultural component of that as well. Humans could have brought some of those bones in there. They could have worked the bones. They could have eaten them. That's another interesting component of it. So, uh, think about our effects of the cave environment. As I always follow the decon protocol, and now we're probably going to be doing COVID protocol for caving, so that'll be interesting. And also realize, your actions in caves can become part of the record for future visitors and researchers. I was thinking about a slide. I think Bill Steele gave a presentation on some of the caving in the Scapegoat, where there was a rope that was put in there 50 years ago that became calcified, essentially. So yeah, just be aware that whatever we do, we're going to be part of that record in the long term. So yeah, let's try and take care of it. I guess that's the take-home message, and get out and enjoy them and see neat things like fossils in caves. Justin or Bruce, would either of you like to add anything?
One of the things that I always found interesting is that you might see some things that are stuck in a wall, and you might automatically think they're old, but just as you were saying right now, Dan, with a rope calcifying and less than a century, it depending on each cave environment, you're going to have a different type of burial for each bone. So it might be cemented relatively quick, especially in a high moisture cave.
You bet, and we'll be happy to take some questions. I guess I would add again that whether the state of the bone, whether it's something cemented into flowstone, whether it's something deeply buried or something on the surface of the cave, I guess kind of the question is it a fossil or is it just an old bone? The answer is yes, and the answer is also it really doesn't matter. Something I wanted to hit on again real quick is that we're seeing a lot more researchers are interested in what we used to call quaternary or recent bones that are tens or a few hundred years old. They're as interested in that as they are bones from the Pleistocene because, of course, there's such an excellent record of climate. So they're all important, whether on the surface, buried, or in flowstone.